look at transcription and translation today. We'll end up down here at the bottom describing exactly what's happening, but I want to do a review over some stuff up here at the top. We'll talk about comparing DNA and RNA, and then we'll end with transcription. So what we talked about yesterday is that you've got DNA, and it is, if you were to zoom in on a chromosome like this, if you were to zoom in on this, you would see a double spiral. And in this case, what we have taken a snapshot of DNA being split apart. And so typically, again, DNA is a double helix. There's two strands, but this helicase enzyme is coming in and it is breaking apart this DNA. It's unzipping it. And so after it's been unzipped, RNA polymerase can come in and it can help turn this DNA into mRNA. And it's not actually changing the actual code of the DNA, it's making not a copy, we call it it's transcribing that over. So if you'll notice, A was paired with T, C was paired with G, but now because RNA does not have any T's, it only has U's, we're doing A is being transcribed to U, G is being transcribed to C, C to G, and T to A. And because now mRNA, this molecule, and it's, it would normally be a longer molecule with lots of base pairs, but in this case we're just taking this one CGA. This mRNA would work get its way out of the nuclear membrane, or through the nuclear membrane, out of the nucleus, and it would come to the next type of RNA in our process is rRNA, and it would come in here to rRNA, like UUU has already done this codon, these three sets. This codon has already done this. Now GAC is sitting here, and what will happen is its pair, the tRNA that will match with this, it'll come over here, sit come to the ribosome, and the thing that the tRNA is holding will then be let go, and then the tRNA can go off and find another amino acid. So I just said a lot of things there. We've got rRNA, looks like when we draw it kind of like a hamburger bun, and we've got tRNA. This is the transfer or transport RNA. The transport RNA is carrying amino acids. This one is carrying a specific amino acid. This one is carrying a specific amino acid, and this one is as well. And we can see that these are different because we see these anticodons, these letters at the bottom of the tRNA. We draw them as CUG, and this one is AAA, and this one is GAU. And so what happens is they will come over and they will drop off this amino acid if the anticodon, these ones at the bottom, match their respective base pair. And in this case, it does. We've got C matches with the G, A matches with the U, and C matches with this G. And so what has happened is this has come over here. It has connected. It has that will release the amino acid, this one amino acid, which eventually, after this happens time and time and time again, it will make a polypeptide chain. You see, it's kind of hard to show this as a snapshot, but GAU worked its way over here, and it matched this CUA. So we have a line of succession here. We've got the UUU matched this AAA earlier on. Right now in this picture, we see GAC matching this tRNA, and then this codon will match this codon, and this red amino acid will pop off onto here. And so this will happen over and over and over until the polypeptide chain is long enough, it's got enough amino acids to do what it needs to do. So you've got three base pairs match up these other three base pairs. This is called a codon when it's on the mRNA. It's called an anticodon when it's on the tRNA, and that is how the amino acids get built into a polypeptide protein chain. All right, so let's talk real quick about DNA versus RNA. DNA is made of deoxyribose sugar. That's the D in DNA. The R in the RNA stands for ribose sugar. So they both have sugars. One is deoxyribose, one is ribose. Let's talk about base pairs. Base pairs, DNA its base pairs are A's and T's and C's and G's, whereas RNA is A's and U's and C's and G's. This uracil, we don't see this in DNA, we only see it in RNA. Then we get to where DNA is, its location is in the nucleus only, it's only in the nucleus. And RNA can be in the nucleus, it can be all over the cytoplasm, so we just write all over the cell. We can see it here, it's in here for mRNA, it's out here in the ribosomes as rRNA, and then going around getting amino acids and transporting them to the ribosome as tRNA. And the shape, the structure of it, it is made of, I misspelled double, that is a double helix. There are two strands there. It is a double-stranded structure, and we can see that when we draw these two lines, that is a double helix, whereas a RNA, I was writing very quickly apparently, a single strand. RNA is made of a single strand, and so it looks like half of that DNA. And so you can see the orange underlines are the similarities. Both sides have A's and C's and G's. Both sides can be found in the nucleus, and both sides are made of strands. All right, last but not least, let's talk about what transcription and translation are. Transcription, we mentioned this yesterday. Transcription is when DNA is turned into RNA. DNA to RNA to specifically mRNA, and this happens right here in the nucleus, is when this is transcribed, when the A is not copied but transcribed into U, G to C, C to G, and T to A. That is called transcription. You can see that example here. ATC would be transcribed into UAG. Now, if I asked you to replicate the DNA first, then that would change everything. So replication is you need to take heed to the words that are being used. Replication of the DNA is going to be a copying of that DNA. So the replication of this DNA would be T, A, G, C, T, G. That would be replication. If you're going to transcribe this DNA, that's going to be what you see here below. And then the next step after transcription is translation. The word is the same, trans. We have trans in the beginning. The first letter that's different is the L and the C. And so these are in alphabetical order, if that helps you remember which one is which. To scribe means to rewrite, so you're just rewriting this, whereas translation is you're changing it more drastically. So this mRNA, UAG, CUG, is this mRNA leaves the nucleus, comes out here, and with the help of a ribosome and a tRNA, or a lot of tRNA, these codons, these groups of 3, are turned into a specific protein. Now in this example, we were choosing random letters, and when we use the codon wheel, we find that it's a stop. That should actually be at the back end. But your goal, what you need to understand, is that the DNA is turning into RNA. As it leaves here, that RNA is then turned into a polypeptide protein chain. It's turned into an amino acid, which is what you've got here, stop and a loop, and then that amino acid is then turned into a polypeptide chain. It's turned into a protein chain.